of Adiutrix, and we are developing first-in-class therapeutics for tissue remodeling diseases. The company is based in technology developed by Randy Silver, who's a professor of physiology and biophysics at Weill Cornell, and she's been studying fibrosis and related mechanisms of fibrosis and tissue remodeling for over 20 years, and has multiple publications and patent filings on mechanisms of fibrosis and prevention. I'm the CEO. I've spun 7 companies out of academic labs over the last 20 years or so. So 1 IPO, 4 acquisitions, 2 approved drugs, and multiple research products still being sold. 2 other members of the team are Mark Junowitz, who's the CFO, and George Kelishvili, who's also a faculty member at Weill Cornell, who's working on the structural biology aspects of our project. So tissue remodeling diseases, they're caused by abnormal accumulation of the extracellular matrix, the structural component of different tissues and organs. And this abnormal accumulation results in pathological changes in tissue structure and degradation or loss of organ function. And this affects many organs throughout the body: skin, liver, lung, eye, kidney, and so on. And Randy's lab has demonstrated that in addition to the canonical TGF-beta signaling pathway that everyone else understands, there's at least one other independent pathway that controls aberrant and pathological extracellular matrix synthesis. Now, most other companies working in this field target only the TGF-beta signaling pathway. And it should be obvious that if you only target that and leave the other one untouched, then that other pathway can still function. But we are targeting a downstream intracellular enzyme, collagen prolyl 4-hydroxylase, or CP4H, and both of the pathways converge on this, and this controls the synthesis of extracellular collagen, which is a major component of the ECM. Our lead molecule is orally available and is demonstrated efficacy with biochemical, structural, and functional assays in multiple animal models of diseases affecting multiple organ systems. And we're currently in lead optimization, and we're generating novel molecules with some micromolar activity in cell-based assays and the requisite specificity. Here's a cartoon version of what I've just said. There are 2 different pathways that direct functional triple-stranded collagen synthesis, one driven by TGF-beta, the other driven by mast cell mediators. Most people focus on blocking this pathway here, but that leaves this pathway still working, and this still drives aberrant collagen synthesis. We are inhibiting this enzyme here, downstream of both these pathways, and so we block the formation of the functional triple-stranded collagen helix, and the single-stranded collagen, pre-procollagen helix, so the single-stranded molecule ends up being sort of discarded via the proteasome. Our lead indications are all pulmonary indications. The first is IPF, idiopathic pulmonary fibrosis, which affects about 300,000 patients in Europe and the US, with an expected lifespan after diagnosis of no more than sort of 5 years or so, and there are more than 50,000 deaths each year from this disease. There are no truly effective therapies other than a lung transplant. Bronchopulmonary dysplasia affects approximately 15,000 premature infants in the US annually who have received supplemental oxygen to compensate for their underdeveloped lungs. And there are no approved therapies for this at present. And it's been observed that as infants who have been treated with supplemental oxygen develop IPF and other pulmonary disorders in their early 20s as a result of this supplemental oxygen treatment. So we ought to be able to prevent that as well. And then finally, pulmonary arterial hypertension. This arises because the blood vessels are constricted through the overproduction of extracellular matrix. This raises blood pressure and increases cardiac effort. There are roughly 80,000 patients with this disorder in the US each year, and there's no known cure, only therapies that control symptoms. So this summarizes a lot of data with a lot of different methods from a lot of different experiments, a lot of different animal and in vitro experiments. And you see at the top, in the untreated IPF model, lung function goes down. And in the lung and other tissues, all of these other measures go up. And we're looking at functional measures, we're looking at biochemical measures, we're looking at histochemical measures, a whole variety of different measures here. When we treat mice with our lead candidate, ADX1001, lung function normalizes and everything else goes down, and in some cases even normalizes as well. I'm only going to show you a couple of pieces of data, but this is looking at the effect of the lead molecule 1001 in the gold standard mouse model of IPF, where mice are treated with bleomycin instilled into their lungs on day 1. And then from day 7 to 14, they're treated every other day with drug or with saline. And then at the end of that time, the lungs are excised. And on the first panel on the left, we're looking at lung function as measured by static compliance. You can see that in bleomycin treated mice, the static compliance goes down. The lungs are a lot stiffer. And then when we treat these mice after bleomycin treatment with 1001, then lung function increases again, and to where it is essentially the same as it was in the control animals. In the center panel, we're looking at the same mice, and you see the bleomycin so pretty much doubles or almost triples the level of collagen in the mice of these lungs as measured by hydroxyprotein content. And then when we treat with subsequently treat with 1001, then again the collagen levels are normalized. Then if you plot the lung function against hydroxyprotein for all of these mice, you get this chart on the right. And this shows that as hydroxyprotein increases in concentration and in amount in the lungs, lung function goes down. And this is just as you see in IPF and just what you'd expect in a progressive disease. So we've worked with 1001 and a couple of other molecules and have two new molecules, which we refer to as 666 and 664. And here we're looking at their activity in cultures of human lung fibroblasts, which have been stimulated with TGF-beta. You see from the Western blot on the top that on the left there's a control untreated fibroblasts, and they have very low levels of collagen. Simulate with TGF-beta and collagen increases tremendously. We have a couple of control compounds there, but then if you look at the dose response curves on the bottom, you'll see that the IC50 in cell culture for 666 is about 86 nanomolar, and for 664 is about 59 nanomolar. You'll also see in the lower Western blot that there is another important enzyme, which are important protein, which is affected by proline hydroxylation, and that is HIF1 alpha, hypoxia-inducible factor. You see inhibiting collagen synthesis has no effect on HIF1 alpha levels for 666, but with 664 we do see a little bit of HIF1 alpha appearing at very high concentrations. So these are as I mentioned, cell culture models, which means that for pure enzyme we're probably looking at 20 to 25 nanomolars of concentrations, which are pretty good and probably as good as we can get. There are two compounds, two drugs on the market for treating IPF right now. OFAB is sold by Boehringer Ingelheim. Esbriet just went off patent a year or two ago, and that's sold by Roche. This is a complicated slide, but the important thing to note is that at higher concentrations, pirfenidone is cytotoxic. So even though at high concentrations we see the level of collagen going down, that's because the cells are dying. And nintedanib is sold as an oncology drug for treating lung cancer. So it's not surprising that we see this being cytotoxic. And then if you look at the far right-hand side, looking at pirfenidone, you see that even at 1 millimolar pirfenidone compared with 28 micromolar, so 1001, the levels of collagen are still very high. So we think that, at least from these experiments in vitro, our compounds are safer than nintedanib and more potent than pirfenidone. So where do we stand right now? Well, we were awarded a CAT grant Q1 of last year, $75,000 plus a $75,000 match from the company. We were also awarded a $2.5 million fast track phase 1, phase 2 STTR in Q3 of last year, and we're on track to meet all the aims of the phase 1 by the end of the second quarter this year. So, assuming that certain organizations in Washington, D.C. behave, we ought to be looking at the phase 2 beginnings of Q3 of this year. We're also accepted at the Ford Incubator at Rockefeller University. We have a couple of desks there, and we'll be moving some more lab work in there towards later this year. And we've accomplished this work working with a number of key CROs. We've a company called Genesis Drug Discovery and Development, or GD3, has been our chemistry partner. Scepter Biopartners, which is also in New Jersey, is working with us on assay development for high throughput screening. And we've also begun working late last year with Schrodinger on in silico molecular modeling for drug discovery and drug design. So we're in lead optimization. Considering the progress we've made already, we think we're pretty much on track to nominate a clinical candidate towards the end of this year, maybe the beginning of next year. The phase 2 STTR grant will fund preclinical pharmacology and toxicology studies for that, non-GLP. As I mentioned, we're on track to achieve our phase 1 STTR milestones, which would mean phase 2 funding will begin in Q3 and Q4. And I should also mention that we have quite a sizable library of analogs of 664 and 666 available now for study. And the SAR data we'll get from that should allow us also to file patents on these new chemical entities towards the end of this year. And with all of that incited towards year end, we're looking to initiate investor meetings Q2 or Q3 of this year. Thank you.
Thank you. And I'm glad we were able to get the presentation back on track. Ron will be at, I believe, table 3 on floor 2 to answer your questions. I think we could take one question from the audience right now while we transition to our final startup, if there are any. Otherwise, please look for Ron after in the networking portion to dive deeper. Okay, I guess, Ron, just in interest of time, so we can keep our last speaker on track.
Yeah.
Thank you for joining, and see you on the floor, networking floor. All right, our last speaker today is Evi Giannakaku.